Scientific discovery has come a long way since 335 BCE when Aristotle proposed that the heat of the male partner during intercourse determines the sex of the child. In 1905, Nettie Stevens studied mealworms and observed that female mealworms have 20 large chromosomes, while male mealworms have 19 large chromosomes and 1 small chromosome. She concluded that the small chromosome, later known as the Y chromosome, was responsible for maleness. It wasn't until the 1920s that Stevens' conclusion became widely accepted. However, her conclusion was thrown into a loop in 1955 when Dr. Sawyer treated 2 phenotypically female patients who did not menstruate and later discovered they expressed the male genotype. This was caused by what we now know as sex reversal, a process wherein the sexual phenotype is redirected during embryonic development. We'll get back to Dr. Sawyer, but first, what is the normal mechanism of sex determination and where does it go wrong?
In 1959, the region of the Y chromosome responsible for the process of sex differentiation was discovered, and in 1990, the SRY gene, the male master gene, was identified as a specific gene in this region responsible for this process. In genotypic males, the expression of the SRY gene results in the synthesis of sex-determining region Y protein. This protein acts as a transcription factor and upregulates the transcription of testes-specific genes, but before it can do so, it must enter the nucleus. How? Nuclear transport. Nuclear transport is a highly regulated process which involves nuclear import and export across the nuclear envelope. First, in the cytoplasm, the transcription factor is recognized and bound at its nuclear localization signal, or NLS, motifs by important beta 1. All nuclear proteins have an NLS, and these are vital for gene expression. Second, important beta 1 interacts with nucleoporin in the nuclear pore channel to transport its cargo into the nucleus. Third, in the nucleus, most RAN at GTPase are in their GTP-bound state. RAN-GTP binds to important beta 1, which reduces its affinity for the transcription factor, and the receptor cargo complex dissociates, leaving the transcription factor in the nucleus.
The ultimate transcription of testes-specific genes stimulates the indifferent gonad to develop into testes. The testes produce certain hormones which lead to the development of other male reproductive organs. In the case of male-to-female sex reversal, the 2 NLS motifs at either end of the SRY transcription factor are mutated. This results in decreased recognition by important beta 1, limiting the nuclear transport of the transcription factor. Less transcription factor is then available to bind to DNA, which results in the downregulation of testes-specific genes. Female-to-male sex reversal is also possible. However, it is not associated with nuclear transport deregulation and is instead characterized by an XX individual's development of male phenotypic traits.
In a clinical context, deregulation of nuclear transport can result in the development of conditions that relate to sex determination. Back to Dr. Sawyer. What is Sawyer Syndrome? Sawyer Syndrome is a rare disorder characterized by the failure of sex glands to develop. This is the condition described by male-to-female sex reversal, where individuals that are genetically XY develop female external and internal genitalia. It is usually discovered that these girls lack ovarian function and therefore do not have sex hormones, such as estrogen and progesterone. As a result, women cannot undergo puberty or get pregnant naturally. Other clinical manifestations include a tall figure, a small uterus, and no sexual characteristics.
When individuals experience these symptoms, hormone tests and molecular genetic testing, for instance fluorescent in situ hybridization, is often used to determine whether one of the specific gene mutations that are associated with Sawyer syndrome are present. 1 in 80,000 people will be diagnosed with Sawyer syndrome in their lifetime. Fortunately, thanks to modern medicine, many treatment options, such as hormonal replacement therapy, are now available to enhance the lives of people living with this condition. Additionally, women with Sawyer syndrome can now become pregnant and carry to term through the use of donated eggs. Our current understanding of sex determination is a far cry from Aristotle's theory in 335 BCE. Since then, we have discovered that chromosomes can differentiate a person's sex and that sex differentiation is highly dependent on nuclear transport processes. As our understanding of these mechanisms continues to improve, so can our ability to design more effective treatment methods.